I'm going to talk about how we deliver adequate, effective, suitable ventilation in the current environment. This is going to be a slightly different presentation to those you've heard because I'm not talking about research that's been done. What I am going to talk about is work that SIBSE has done over the last few months. We became aware, and by we I mean senior members within SIBSE, particularly those in our heating, ventilation and air conditioning interest group, and also within the natural ventilation group, became aware that aerosol transmission did appear to be a prospective pathway, perhaps a little bit ahead of the World Health Organization and others, and perhaps in line with some of the others that we've heard speak today. And we issued guidance initially in early May, which has been revised a couple of times, that was aimed at helping people to get back into buildings. And the scale of the challenge is that in the UK, we have something like 2 million non-domestic buildings. And many of them are naturally ventilated, others will have mechanical systems, some will be air conditioned. But if we're going to return to work effectively, then we need to reopen those buildings and we need to enable the people who run them to be confident that their ventilation systems are reasonably safe and are minimising the risks to the people that they're asking to come back into those buildings. So that's our overall target. And what we're trying to do, as I think everybody appreciates, we're trying to reduce the potential concentration of any viral material in the occupied spaces. And the easiest way of doing that is by increasing the air supply to a space. Now, I think we all appreciate that exposure depends on the number of occupants in the space. And as we've just been shown in such a vivid way, the levels of viral load are going to depend on the exertion that's going on. So sedentary occupants talking quietly on the phone are clearly going to generate lower levels of viral material than somebody who's walking around the room and talking loudly. And that's obviously going to affect the levels of viral material that need to be diluted. So what are the challenges? Well, earlier presentations have shown us that there are quite a number of significant unknowns at the moment. And bearing in mind when we were producing this guidance back in the spring, I'm not sure that everybody expected to be where we are now and facing the length of time with ongoing restrictions of the scale that we now face. But there are some other challenges as well. We don't know the location of any source of infection because the biggest problems or the biggest risks are those who are pre-symptomatic or asymptomatic. And by definition, they don't even know who they are probably, let alone those around them. And they're going to be moving around and shedding viral material. We don't know the rate, and we've seen a lot of evidence this afternoon about the rate of infection and how that can vary. And so some people are going to pose a bigger risk than others, but we're not going to know who they are. And clearly the behaviour of infected material is being influenced by the movement of other building users. And for those who weren't able to hear Andy Woods earlier, I would recommend having a look at his slides because that shows very, very clearly that movement around buildings is going to have a significant influence on what we're trying to address. So those are the key challenges. What we're currently recommending, and bearing in mind this is aimed at the vast majority of people running the 2 million non-domestic buildings in this country. And many of them have very limited technical expertise. So this is about how we provide straightforward advice to probably tens of thousands of building operators. And so clearly we need to encourage them to understand the ventilation system. If it's a naturally ventilated building, that may just be the windows. But how many people know how to use sash windows correctly, to open top and bottom so that you get proper air change? They may be old-fashioned, perhaps the knowledge of how to use them has gone as well. For those who've got mechanical ventilation systems, clearly we're encouraging people to run their ventilation at higher flow rates, and this may need people to change their CO2 set points where those have been used to control the ventilation rates. We have been encouraging people to avoid recirculation. Again, the reasons for that have been made very clear in some of the earlier presentations, and I think it was very interesting to listen to Clive Beggs talking about the potential use of UV in return ducts. And again, Clive was at pains to point out that this needs further exploration, and I think that there will be people who are beginning to look at that. But at the moment, recirculation of air runs the risk of recirculating infected material. That's not the same where you've got recirculation of air within a single room, as long as you've got a source of outside air. Apologies that the word fresh air crept onto that slide. Outside air needs to get in so that there is a mix of outside air and what's being recirculated. So in summary, quickly, we see increased ventilation using outside air as the primary means of mitigating the risks of indoor aerosol transmission of the virus. That's not to say that other measures are not relevant or appropriate. Indeed, things like hand washing and cleaning of surfaces are clearly very important alongside that. We do see and have been saying for some time that recirculation does increase the risk of transmission. But we're also very aware that winter is posing a challenge of balancing adequate supply of outside air and thermal comfort, and that is going to require some further judgment. And there is clearly a need to think through further the potential for other approaches, and we've seen reference to air cleaners and to UV in other presentations. We need to do some more work to establish how realistic it is to use them and how they should be used, and what sort of guidance is needed on installing them. So that's a very quick summary of the guidance that we've been putting out, and I understand from my colleagues that we've had about 35,000 downloads of our guidance on reopening buildings, which shows it has sparked a reasonable degree of interest, and I'm also pleased to say it's been picked up by the Health and Safety Executive. So with that, we're taking questions at the end of the session, not now. Thank you very much for listening, and apologies with whatever gremlin it was that caught me at the beginning.
Great, thank you very much. So yeah, again, do put your questions in the question box, and we'll have a discussion session at the end. The next talk is Dr. Ginny Moore from Public Health England. Ginny, whenever you're ready.
Okay, so can you see my slides?
Yes.
Okay, first hurdle leapt. Okay, so just by a way of a very brief introduction, I'm part of the biosafety group based at PHE Porton Down. We are an applied research group with a particular interest in the role of the indoor and built environment in the transmission of infection. And over the past few months, we've been carrying out lab and field-based studies to investigate if, how and when people with COVID-19 contaminate their surrounding area. And this has involved us taking surface swabs and air samples. And so over the next few slides, I'm just going to give a very brief overview of some of the work that we've been up to and some of the challenges that we faced. So understanding how SARS-CoV-2 is spread within the hospital setting is obviously essential if staff are to be adequately protected, if effective infection control measures are to be implemented and nosocomial transmission is to be prevented. So as part of the PHE instance response and over the course of the outbreak, we visited 8 hospitals, some on more than one occasion, and investigated the presence of SARS-CoV-2 in the air and on environmental surfaces around patients with and without respiratory symptoms. Our sampling primarily focused on individual bed spaces in negative pressure isolation rooms, neutral pressure single rooms, and cohort bays, but samples were also taken from the wider ward environment and from non-COVID wards. All samples were returned to Porton, where they were analysed using PCR and virus isolation assays. Now, ambient temperature and relative humidity differed with ward and ranged from 21 to 25 degrees centigrade and from 21 to 41% humidity. Now, I'm not going to talk about the surface sampling today, but in terms of the air samples that were taken, these were collected less than 1 metre from the head of 44 different patients, 21 of whom were receiving mechanical ventilation, either invasively or non-invasively. 6 patients were receiving oxygen via a Venturi mask, and at the time of sampling, 3 patients required drugs or saline to be administered by nebulization. And SARS-CoV-2 RNA was detected in 4 of 55 air samples. In all 4 cases, the concentration of viral RNA detected was low and ranged from less than 10 to 460 genomic copies per metre cubed. Now, in a rapidly evolving outbreak situation, there is, of course, a need to gain a quick understanding of certain trends. In this case, if and how COVID-19 patients contaminate the healthcare environment, and whether this presents a significant risk for staff and/or patients. And the results from our study do support the findings of others and suggest that in wards caring for COVID-19 patients, the concentration of viral RNA in the air is likely to be very low, and that if worn and used correctly, the PPE recommended within the UK, including when indicated components to protect against aerosol exposures, should provide adequate protection against potential virus exposure risks. But of course, our study has its limitations, largely due to the necessary but reactive nature of the response. But I think that we can learn from these experiences and use them to form a more robust research response. So as mentioned, SARS-CoV-2 RNA was detected in 4 of 55 air samples. Now, it's not known what may have contributed to this airborne contamination, but 2 samples were associated with the same make and model of CPAP machine that wasn't used to treat any of the other patients. And on one other occasion, a notable increase in ward activity was observed, and this may have facilitated airborne dispersal. So arguably the result of sampling in the right place at the right time. Now, when sampling the healthcare environment, of course, you must be very mindful of the staff and particularly the patients, and sampling can be very intrusive. And because of this, we chose to use air samplers that could sample a large volume of air in just 10 minutes. Now, whilst this did increase our chances of detecting SARS-CoV-2 in the air at that particular place and time, where and what activities could be captured was, of course, very opportunistic. And it's acknowledged that many of the procedures believed to generate aerosols and droplets were not captured during our study. Now, samplers operating at a much lower flow rate can be used to sample the air over a much longer period. But without observing ward activity, it becomes much harder to interpret a positive result and to determine cause and effect. And I guess it's also important to consider where sampling should be carried out, not only in relation to ventilation and airflow, but also in the vicinity of who. In our study, viral RNA was detected in an air sample taken close to one of 6 patients receiving oxygen. The time since this patient was diagnosed and had experienced symptoms was 1 and 6 days, respectively, which was comparatively earlier than many of the other patients that we sampled around. Now, studies have suggested that the viral load in the throat is highest on or before symptom onset, and so it's possible that the majority of hospitalized patients do not shed high numbers of viral particles into the environment. Now, to investigate if symptom duration could or does impact environmental contamination, we also carried out environmental sampling in the homes of individuals who reported recent onset of symptoms, specifically the loss of smell. And our results confirmed that people experiencing anosmia may, as a result of COVID-19, may expel viral particles from the nose and or mouth, and that low concentrations of airborne virus may be present. Now, that hands and fingers may also be contaminated, and as a result, viral particles may be present on high-touch personal items such as laptops and e-readers and domestic surfaces, particularly those associated with higher contact pressure and grip, like the fridge door handle particularly. And obviously these are likely to be used by other members of the household, which means, of course, household transmission is likely. And just to expand very briefly on case number 3, whilst we were sampling, we were asked by their partner if we would sample their hands as well. We did, and viral RNA was detected. Now, this person was asymptomatic at the time of sampling, but did become ill 2 days later. Now, what we don't know, of course, if this is evidence of fomite or airborne transmission, or if they were already infected and prior to showing any symptoms were contributing to the air and surface contamination. So bearing in mind then that many people could be infected with SARS-CoV-2 and not know it, as we've heard, there is a need to understand the type of activities that could increase dispersal and spread of the virus. But carrying out controlled environmental assimilation studies is difficult, particularly if results are required rapidly. So we've been investigating the use of respiratory bacteria present in high numbers in saliva to determine if and how different activities differ in terms of risk and the effectiveness of intervention and or mitigation strategies. So with the help of PhD Porton staff and choristers from Salisbury Cathedral, we took part in the same study we've already heard about from Dr. Bezdek and assessed how far droplets and aerosols are spread during singing. And the graph on the left shows that significantly fewer respiratory bacteria were recovered during singing than during more dramatic expiration, such as coughing. And during a similar study, a different set of volunteers were asked to carry out a series of respiratory activities, which included coughing, speaking loudly, and singing when wearing a mask, a surgical mask, and a homemade cloth face covering. And the graph on the right just shows that both types of face covering can significantly reduce the number of respiratory bacteria deposited onto surfaces. So is the use of respiratory bacteria an appropriate approach? Certainly the salivary glands are thought to be an early target for SARS-CoV-2 infection, and saliva is of course currently being investigated as a non-invasive specimen for the detection and the quick and easy diagnosis. Now, the concentration of live virus present in saliva is not known. Published data tends to be based on PCR data. But the concentration of respiratory bacteria present in the saliva of healthy individuals is thought to be higher, so conceivably could be considered worst case scenario. Bacteria obviously differ to viruses in terms of size and structure, and so deposition rates could differ. But does this matter? As we've heard, other proxy markers such as airborne particles have been used to assess relative risk, so the number of particles generated during one activity compared to another. And the spray factor, so the proportion of microbial or respiratory bacteria present in the saliva that is dispersed, can be used in a similar way and can be used to quantify the risk associated with different activities. It could also perhaps be used to identify potential super spreader events. Now, certainly not all our volunteers dispersed respiratory bacteria, none of the female volunteers, for example, despite them all having very similar concentrations of respiratory bacteria in the saliva. But while some people may not generate and spread respiratory droplets. Others, for whatever reason, can and to produce high numbers. Now, in comparison to when these volunteers coughed, for example, fewer respiratory bacteria were dispersed during singing, implying a lower relative risk. However, one volunteer produced significantly more respiratory droplets during singing. Why? I'm not sure, but I think these differences between individuals does warrant further investigation. Understanding perhaps the dispersal from the source may actually be easier, may actually help mitigate any potential or the potential effects. So a lot of people involved in this study, so just thank you to them, and I'll take questions at the end if there are any. Thank you very much.
Thank you. Yeah, so please do submit your questions. The next talk is Professor Wendy Barclay from the Department of Infectious Disease at Imperial College London. Wendy, whenever you're ready.
Hello, good afternoon. I'm hoping that you can see my slides and hear me.
Yes, all looks good.
Great, okay, well thanks very much for inviting me. I'm a sort of academic virologist with an interest in the way that new viruses emerge from animal sources, and for some time I've been interested in transmission as one of the big barriers that an animal virus has to overcome in order to turn into a pandemic. We used to study influenza, but these days we spend quite a lot of our time thinking about SARS-CoV-2, but I'm going to show you the kinds of approaches that we take to make these studies happen. So I think we've spent a lot of time today on things like environmental factors and probably somewhat less on the virus characteristics that determine whether or not a respiratory virus is going to transmit well between people. How well does it replicate in the cells of the human respiratory tract? Can it find the right receptors to enter cells? And importantly, how stable is it outside of the host? Does it retain infectivity during the sort of risky period for a virus? So I think of things in this way. This is a picture of influenza viruses emerging from an infected cell. This could be perhaps the cell of an infected person in their nose or throat. And there are all these viruses which are going to be caught up in respiratory droplets and exhaled or sung or breathed into the environment, from where they'll need to enter the host, make their way through the mucus barrier, which can be quite a problem for some viruses, and then find target cells below in the ciliated epithelium to initiate infection in the new host. This is a pretty hostile environment for a virus, so how on earth they... We would love to study all of these things in humans, but it's quite difficult to do that. It's quite difficult to find people at the very beginning of their infection. They usually only turn up in hospital or at the doctor's 2, 3 or 4 days in. And that, amongst many other reasons, makes us turn to animal models. And most of our work is done in ferrets, because ferrets are a very good model for influenza. And the way we do this is that we infect a naive animal with a known dose of virus at a known time. We introduce another animal either into the same cage to mimic a sort of very close setting like a household or a classroom, or we perhaps introduce an animal into the adjacent cage where the air can pass between the two, but of course there's no physical contact, and this mimics very much transmission over longer distances. And the sort of data we get, well, every day we nasal wash the animals to wash out in a buffered solution the amount of virus in their nose, and we record that by plaque forming units, how much infectious virus is present in the nose. So if we take 2 ferrets and we infect them with the pandemic flu of 2009, you can see that they shed virus very early on, day 1, and peak out at day 2. If you're measuring clinical signs in these animals, the first clinical signs you'd see are not until the evening of day 2, when they show a fever. They then might start to cough or sneeze around day 5, 6, 7, or even later. But after day 7, they've cleared the virus and there's no infectious virus in their nose anymore. So any symptoms or clinical signs that occur after this are kind of irrelevant to the virus. Placing animals in the same cage on one day after infecting them is a very efficient way to pass the virus from the first donor animals to the contacts. Placing animals in the adjacent cage is less efficient route for transmission, and we can show using genetic studies that the number of virus particles that initiate this infection is many fewer than the number of virus particles that initiate the infection in the case of the direct contact. This is our first set of data really with SARS-CoV-2 using the ferret as a model in the way that we've done with flu. This obviously has to be done with precautions as appropriate in high containment. And here are 4 animals in orange, nasal washed every day, and the amount of virus in their nose is recorded either by RNA copy numbers or infectivity. And you can see that in 2 of these 4 animals infected with wild-type SARS-CoV-2, we had a direct contact transmission event. There are 4 other animals shown here in pale blue. These are infected with a cell culture adapted mutant virus of SARS-CoV-2. And you can see that although they become infected, they actually shed less virus in their nose and they don't actually transmit onwards. So from a virology point of view, this can give us great clues about the features of the virus as it's evolved from animals into humans that's enabling it to transmit amongst mammals as these ferrets are models for. Jay is my postdoc who does all of this work with us, and he was previously employed in Hong Kong where they had a very nice setup where they wanted to address the size of the respiratory droplets that were carrying the virus from the donor animals to the recipients. And so they used an impactor to only allow through respiratory droplets of certain sizes. And here you can see with a typical well-transmitting influenza virus that it's the larger droplets which seem to be more important, if you like, for transmitting the infectivity from the donor animals to the recipients. And when you get down to very small droplets of 1 micron or less, then really you don't see any transmission. We've got an apparatus which is much cruder than that downstairs and will make many people on this call cringe, I'm sure. But this is our apparatus to basically measure virus in air that's emitted from an infected animal. And we simply open the lids of these plates and allow droplets to fall onto these plates. We can then directly quantify the infectious virus which is being emitted from an infected animal. And we can use that to look at things like how many days after infection is most virus emitted. This we think is important because it's very important to understand if people are more contagious to others early or late in their infection. Just symptoms really contribute largely to the transmission, or is transmission more likely to happen in the asymptomatic or post-symptomatic time. Lara very kindly modelled for us what she thought would happen to the droplets emitted in our tube. And we think that we're picking up deposited droplets in our plates of 7 microns or larger. And if we look and see, does that droplet deposition on the plates carry infectivity? It does. But what's very interesting is that the very peak of infectivity emitted from the infected animals in our case is on day 2, when I've already told you the animals are not showing any clinical signs at all. So in this model, we see that the emission does not necessarily correlate with how much virus is in the nose of the animals, but it very early on. And indeed, if we only expose sentinel animals at this early period, we get very efficient transmission, whereas if we expose sentinel animals much later, we don't see transmission at all, even although the donor animals have got lots of virus still in their nose. So the notion that coughs and sneezes really spread all the diseases, I don't want to say they don't, but I think there's plenty of evidence listed here that a large proportion of transmission, both of influenza and probably SARS-CoV-2, is not dependent on symptoms, but can happen in the asymptomatic or post-symptomatic period. And why might we see less transmission as symptoms proceed? Well, we think the contents, the quality, if you like, of respiratory secretions changes over time. And as the virus is exhaled in respiratory droplets, the content of those respiratory droplets may well concentrate innate factors, including ions and mucus, which can have quite adverse effects on the infectivity of the virus particle. For example, we know that the pH of the human respiratory tract is already mildly acidic, but it becomes more acidic during infection. And some viruses are exquisitely sensitive to pH. To illustrate that, we engineered 2 influenza viruses that were differentially sensitive to pH. This orange virus has a point mutation in it, which makes it fall apart if it hits pHs of 5.5 or less, whereas this other virus is a wild type virus. If we infect our animals with these 2 viruses, they both get equally infected and shed virus over the same period of time. But if we collect the emitted air from the animals, there's no infectivity emitted from the animals infected with the unstable virus. Whereas, once again, on day 2, we see plenty of infectious virus in the air emitted from these animals with wild type. And we think what happens is that the unstable virus, as it's emitted in these respiratory droplets, which are already mildly acidic and full of ions and mucus, becomes inactivated as water evaporates and these droplets become smaller and concentrate their constituents. So I'm conscious that my talk is very different than what you've already heard. The big question for me as a virologist is, why does this SARS-CoV-2 transmit so well, and in particular in comparison to SARS-CoV-1? And I think the real clue is about transmission efficacy early in infection. Both of these viruses are stable in the environment. They're both well stable in air. But the key to being a pandemic virus is being able to transmit from people who are not even detected as being infected. So I'll leave it there, and thank you for listening to me, and look forward to having any biological style questions from you. Thank you.
Great.
Thank you very much, Wendy. That was fascinating, and I think the difference between other talks is greatly appreciated. We now have a representative from the Department for Transport, and Asha Lawrence Cole is going to speak next in that capacity. Over to you, Asha.
Thanks very much. My name is Asha Lawrence Cole. I'm joint leading the Department for Transport Science Response Team with my colleague Dan Achampong. I'm here to speak about COVID-19 and transport, to give a perspective from the science at the policy coalface, and to discuss some of the challenges we face, some key questions, and briefly give an overview of the TRACK project, which is designed to address some of these challenges. Let's see, there we go, it's working. So transport is a key sector for reopening the economy. I'm sure you've all seen the news stories which have been associated with this. It's a really high priority area to be able to do such things as kind of return to school, and more recently the government ambitions to return people to offices. I mean, COVID-19 has had a huge economic impact on the transport sector. Tens of billions of pounds of kind of economic damage, and kind of the scale of this is brought kind of science to the front line in policy making. We have to say that transport wasn't necessarily particularly well represented before now in the kind of overall understanding of pandemics, but that's really turning around. We've had to take a really fast-paced approach to bringing evidence to policymakers, such as in the effort to make transport the first sector to mandate face coverings. So this has all been enabled by a really close relationship with, say, GMG, with many members of which will be here, I'm sure. The work done early on in the crisis by that group has kind of highlighted for us the key risk factors as shown here, which are common to many environments, but they're often uniquely present for transport. Especially relevant to the interest of many researchers here is the factor of how enclosed many transport environments are, and the complexities of the ventilation systems within those environments. There's kind of unique challenges presented between cars, buses, trains, and planes, frankly. I suppose one risk factor we've got to count ourselves as lucky, as we've seen from the other presenters. We're lucky about how reserved we are about having conversations on public transport. So there is something going for us in this country. So aside from that, the policy demands for research on COVID-19 have been enormous. This slide should highlight some of the journey that we've taken in informing policymakers from the kind of absolute fundamentals of infectious disease and public health to having specific and detailed questions coming out of all of the policies and the questions and the issues that have been faced by policymakers. So some of the kind of really challenging questions that have come out is, for example, what is the impact of sitting with different orientations? Because we know that there's, it's better to sit not facing others, but understanding quite how directional the aerosol plumes we emit are and what the impact of ventilation on them is. We've seen some really interesting results already presented on kind of which direction and essentially directionality in the aerosol plumes kind of through carbon dioxide sensors on trains, but how that kind of works across public transport modes, and kind of trying to understand, is there a seating arrangement that's safer than others? Is there a particular way of sitting that would be really useful? Understanding how we understand that the risk factors around the number of contacts from outside your household that you come into contact with, and the length of time spent in contact with them. Kind of understanding the linearity or non-linearity of that relationship is kind of a really key, interesting thing for us right now. And then about face coverings, they've been mentioned throughout the day. And I'll be clear, I mean face coverings as mandated on transport rather than masks or respirators, because there's a really key difference. I think we've seen that today. There's a growing evidence base for these, but what is the kind of relationship with actual policy compliance of people on transport? So I'll use a controversial term here, but we know that there's a potential of some kind of herd protection effect with people using N95 style kind of respirators, but that's really looking at kind of individual protection. But face coverings are more considered as kind of a source control method. So actually, how does that relate? And kind of, are you looking at diminishing returns to get the last kind of 5 or 10% of people on transport wearing face coverings? Another thing is around, kind of how do we quantify the impact of mitigations that are in place on transport, and understanding risk as a comparison to other factors. If we say public transport is hugely dangerous, don't go on it, which obviously we're not saying right now, then how do we compare that kind of saving in COVID risk to other risks such as road deaths or risks on health from decreased mobility? It's kind of a hugely challenging set of research questions, and I have to say this is only really coming from my perspective as more of a physical scientist here. There's equally many in various other areas such as the social behavioural field and the economics field. So I think the biggest question which I'd like to highlight here is on understanding the evidence for transmission on transport. It's an area we don't have a very clear answer on. Is transport a high-risk environment? How much do we have to worry about increased use, and how do we minimize transmission on transport? SAGE EMG has brought together a lot of evidence around this, but there's still kind of a good few questions around that. So other routes kind of outside of the understanding of environmental transmission that can add to the same question, such as kind of understanding how we better do contact tracing on transport using technology, for example, and looking at the risk of transport users on a statistical level. So these questions have led kind of DFT, CSA, and SAGE EMG to pull together the TRACK project, which is being led by Kathy Noakes. This is one of the most significant projects to be funded by UKR on COVID-19 research. I'll give a brief overview here. So TRACK is an 18-month project starting in September, which the department is driving to work in a somewhat agile way, bringing early data and modelling to policymakers as rapidly as possible. I'll be quite honest, policymakers, they look for answers, within days and weeks rather than months and years, as we know the science takes. It's worth saying, though, that many of the work packages are actually already going ahead. So work package 1 is bringing together all of the understanding of specific routes of transmissions gained in the TRACK project and elsewhere to build a risk model. I won't say too much of this because I know that Simon immediately following me will be saying it in a lot more detail. But this is the central core of the project, which the other work packages are feeding into. Work package 2 focused on surface sampling. You might imagine that Ginny is involved in this. So that's to understand levels of surface contamination and really will inform our understanding of fomite transmission on transport. Work package 3 focused on user behavior to understand kind of the who, when and why of transport users. Will enable us to understand better the risk factors and demographics of the traveling public. Work package 4 focused on CCTV analysis will help understand the actual behavior of passengers on transport, including distancing and surface touching. This will actually allow behavior to be understood and modeled accordingly. Finally, work package 5, environmental transmission and mitigation, focusing on airborne transmission and mitigation, being led by Paul Linden, which will be bringing together many of the kind of airborne transmission subjects we've discussed today and looking at the specific application to transport. So this slide shows a little bit of the interaction of the work packages to create a kind of a comprehensive picture of environmental transmission on transport. It should be clear by now that the overall question which we have around understanding risk on public transport is hugely complex and interactive between many areas of science. Track is kind of a large part of the picture, but it still is only a part of the picture. So almost all of the presentations today have been of some direct relevance, from understanding what is an infective level of aerosol in the air to the more practical discussions around mitigations. It's worth also saying that track will feed into the kind of the higher level epidemiological understanding of where transport fits in the overall transmission picture across the country. So to conclude, transport is a key sector. It's heavily impacted by COVID-19. The evidence around the risk of transmission on public transport is a key challenge, and track is a significant program of work contributing to answering that. And DFT will continue to work with academia going forwards. Thank you.
Asha, that was fascinating. Thank you, and it's really nice to have someone from Department of Transport telling us the relevant issues there. So we're now going to have a talk from Dr. Simon Parker, following on sort of this theme of governmental department and the relevance to them. Simon is a representative from the Defence Science and Technology Laboratory. And it looks like his slides are there now.
Thanks, Henry. Can you hear me?
Absolutely.
That's great. Thanks for the introduction, and thank you to Asha for setting the scene as well. So I'll lead on from that to talk about one element in particular of the track project that Asha introduced, which is really the risk modelling element, and trying to create a sort of holistic view of transmission risk in transport, and making estimates of the contribution of different routes of exposure within transport environments. So I think I won't dwell on this. I think Asha introduced it. There are 5 work packages within the track project that's due to start on the 22nd of September. It's running for 18 months. And we've got some early deliverables. First one's at 3 months, then 6, 12, and 18 months. And really the work's very much focused on trying to provide a route for all of the evolving science in lots of the different areas that we've heard about today, and that are being developed under the different work packages, to feed into a better understanding of risk. So how we hope to do that is through work package 1, which is about modelling transmission risk on public transport, and it's being led by the University of Leeds, and DSTL and University of Manchester are working with Leeds to develop models that are really split into 2 different parts. We have a stochastic risk model that's designed to simulate the different routes of transmission, to simulate individual journeys, and to stochastically represent passengers as they undertake those journeys with different properties. So whether they're infected, for example, and if they are, how much virus they're emitting. Other behaviours like mitigation measures like mask wearing, for example, but also their proximity and how that changes over the journey. And by developing that microscale model, we hope to build up a picture of the risk of transmission under lots of different transport scenarios. So looking at tube travel, train travel, and bus travel, and then wrapping the results from that microscale model up into a planning tool that's more able to deliver the kind of advice that policymakers can use more directly to help them draw on those insights. And as I said, the work package will draw on the other elements within the project that are looking at surface sampling, looking at how people travel. They're looking at people's behaviour as they travel, so using CCTV to look at proximity, but also touch behaviours on transport, and some of the work that Paul and Chris are involved in looking at more the finer levels of detail of airborne transmission in those environments. So what I'm going to do is just very quickly talk through some early work we've been doing to try and put together a framework that can be used for this kind of microscale modelling, and should say it's very different from kind of high fidelity modelling we looked at earlier. It's not a CFD type model. It's really based around representing those different routes of transmission. So our initial test case is based on London Underground and the Victoria Line. It's based on a single carriage, either going northbound or southbound. We have the details there for the volume and the surface area and the ventilation details, and we're making some assumptions at the moment that we'll update as we get more data about surfaces that are touched, handrails, seat rests, etc. And then the events that happen, so people are boarding the carriage and alighting at different stations, and the length of time that they're on the carriage. And it's obviously an example that's scenario that's of interest, because it's so widely used, and also because the passenger density is so high. But in other respects, it's at one edge of the kind of scenarios that we might expect to see in transport. In fact, the ventilation rate is very high because of the number of people that are carried on the tube. So in some ways, it's a starting point, but we'll use that to build towards other train travel and buses as well. And then the passengers, we'll treat those individual entities. They can get on and get off any station. They've got a particular probability of being infectious, and they accumulate a dose during their journey by each route of infection, and that's what we'll model. So, as I say, we've made an early start on this, but the project doesn't start in earnest until later this month. So I'm just going to cover the methodology very briefly, and then show you some really early illustrative results. So we're tracking 3 different routes of exposure: airborne exposure via small aerosol, and at the moment just in a single well mix zone, but that we're looking to update that as we improve the model. Then close range exposure through droplet transmission, and then the fomite route through surface contact. And what we're doing at the moment is we're not tracking individual locations, but we are tracking the people who are within proximity of an infected individual. So if there's an infected individual on the carriage, we track those within a metre and those within 2 metres. And we adapt in terms of the passenger density and the constrictions due to the carriage to account for how much area is available there. So in the interest of time, I will skip very briefly through these, but our very early sensitivity analysis suggests that source strength is by far the largest variable in terms of influencing the airborne exposure to small aerosol. The ventilation rate, it's very high on the Victoria Line, but we expect for buses and trains that will be much lower. Again, that's another cause of variability. Relatively little variability due to deposition and decay rate. Then as we look at proximity and close range exposure, this plot on the right shows passengers as they move between stations. The red is the red line along the bottom here is the infected passenger. Those in orange are within 1 meter and those in yellow within 2 meters. And as the passenger density goes up, we see that more people fall within those proximity areas until that infected passenger gets off. And we can see that actually how we account for that area makes a difference, certainly to the numbers within 1 to 2 meters. There's not enough area for a 2 meter circle within these carriages. And we can look at surface contact, and again, very early results, but we see the further the train travels along the station, the rate of contamination, the mean of the contamination falls off as it's spread over more surfaces and more passengers, but more surfaces and passengers have some level of contamination. We make some assumptions about how many surfaces they touch, and that's important in terms of there's a point at which it reaches most of the surfaces or passengers have some level of contamination. And an important variable is whether we model whether people just board with contamination on their hands that they then transfer, or whether they cough or sneeze onto their hands. And from listening to some of the talks today, perhaps we ought to think about breathing onto surfaces as well. In the interest of time, I'll wrap up. So we've done some early work in terms of putting together a model and doing some early sensitivity analysis. There's quite a lot of work still to do, and keen to build on the science as it evolves in terms of the close range exposure, better understanding of dose response, whether we use SARS-CoV-1 for the moment or before other data becomes available. And then we plan to do a fuller sensitivity analysis to start to build up towards this planning tool. And further ahead, we'll move into extending to other environments, buses and trains, etc. Ultimately, with the aim of delivering policy advice to DFT, but also to other transport operators, and potentially to read across to other environments. And I'll stop there. Thank you.
Thank you, Simon. And I think, yeah, your final few comments. I think I'm sure we will learn a lot from the extreme high-touch surfaces in transport that will translate, perhaps in a diluted form, to other surfaces. But that will be interesting to see. Steve Wisby, if we are slightly ahead of schedule, but I am aware that we're at the end of a long afternoon or so. So I don't have any shame in starting Steve a little early, if that's okay by you, Steve.
Yeah, of course it is. Yeah, I'm very surprised we're on time. Can you see my screen okay?
Absolutely fine.
Fantastic, fantastic. Right, I am, as Hugh was saying, there are 2 million odd non-domestic offices and properties in the UK, and I look after one of those for our firm. It's actually HVAC, or Building Services Engineers Consultancy, and I'm going to cover 2 things today only within the 10 minutes, and that's the distance issue that we have for reoccupation and dilution, as in the ventilation system. I'm going to start with a quick overview, as in this is the office. It's actually restored a transit shed on the King's Cross estate that Argent developed from the early 2000s, took a long time developing. Our offices are pretty typical, but we've got a nice double height space with desks arranged primarily to suit actually the ventilation as it happens, because the ventilation in this building is served through the underfloor arrangement rather than above, because of the form of the structure. But if I first start with distance and what we currently do in the UK in relation to others, and you can obviously, everyone's talking about the 2 meters, and I've been as an employer and looking after my office following government guidance. And you can see that we are only one of 2 countries that keeps to 2 meters. And 2 meters actually to me is 200 millimeters too much. And I'll come on to that, but 2 meters is where I'm working. And it talks about 2 meters with 1 meter, obviously with 1 meter plus, with mitigation if you're actually working less than 2 meters apart or in activities in the office, which is all fine and well. But then I started looking at the current support and the EMG, which has already been mentioned, about the science behind the 2 meters, and something that I found isn't reported in the government guidance, and I wish it was, because it may actually make planning of spatial fit of people coming back to offices a bit more relaxed or actually improved. And there's obviously guidance about what we've been discussing this afternoon with regards to dispersal and the fine particles and droplets, and this general 2 meter rule, and it actually refers to less than 2 meters. But it also goes on to say in the current EMG, through SAGE supporting documentation for the government advice, that there are the risk of having contamination spread is effectively the same if you're 1 meter side by side or back to back. Now that isn't reported in the government guidance, so at the moment everyone draws this lovely 2 meter diameter, sorry, 2 meter radius around a body, but in reality I think it actually looks like that because it is more directional. And if it went down to 1.8, we could have a lot more people in our offices, a lot more. But 2 meters is where we're at, and that's what we're working to. We're not imposing masks in the offices, so we're sticking with mask-free and 2 meter separation. Now, our office is fairly typical with regards to straight desks. We say are working primarily the distance between the desk is to suit the ventilation which comes up through the floor, which I'll come on to in a minute. So we sit generally at 1.4 meters apart and 1.85. So if you apply those 2 meter diameter radius rules, you suddenly end up with alternate desks, which is not unusual. There's a lot of plans out through interior designers to reoccupy at this sort of density. Yeah. We are installing CO2 sensors. We've got our own in the offices, and I've actually shared that data with Henry already. And as it happens, today, we occupied on Monday. We had an issue where the ventilation failed, and I just thought I'd please Henry by showing the data with regards to the CO2 that started to climb up, but obviously not to very high levels. It's a nice big space, but not many people in the office at the moment. There's only about 10 people in our 22,000 square foot office. But it is useful data, and it will be used and monitored with regards to concentration at perimeter strip. So coming on to the ventilation, the dilution, this is actually through underfloor air supply system. It does in a region of about 7 or 8 air changes at the perimeter and about 4 to 5 internally within the depth of the building. Now, I've been through over the last week or so preparing to get to the offices, and even in engineering environments, you end up with people wanting to shut the things down and actually stuff them up. So I'm glad I did that, because even as engineers, we end up with people shutting off and not realizing how important ventilation is. So we tested this prior to coming in the office, and we looked at the range of air going through the supply air grills. 2 scenarios, one at minimum volume, which is 15 litres per diffuser, and 40 litres per diffuser. It actually, under low minimum volume, acts very similar to a displacement system, where the air will actually track up thermal plumes generated by bodies and equipment. But under 40 litres, it actually ventilates and mixes up to a height of around about 2 meters. So it does the occupied zone. And during the summer of, I think it was 2016, yes, I actually hung some nylon strings with temperature sensors on in the middle of the desks at both levels to actually demonstrate the performance of the system. And I'm glad I've done this, because it's actually found a use in this presentation, because it clearly shows that the ventilation system controls to maintain comfort conditions at low 2 meter height. And as it goes through the day, you can see that the roof lights actually get very warm underneath. They have blinds underneath, but even that, they get up to quite high surface temperatures. And at the end of the day, when the whole system turns off, it turns quite red, primarily because there's no air circulation pushing that ventilation load, sorry, the thermal load up into the space, away from the space. So we've spent a lot of time on our ventilation system and making sure that the SIBSE guidance and REVA guidance is complied to make us COVID secure, following government guidance. We have a system that basically pulls from the space, exhausts, and we've got return air as a means of heat recovery, and supply air with outdoor air. It still actually says fresh. I'm sure you'll be glad that I've crossed that out. So that damper is obviously not allowed. It's been isolated both physically and with its actuated taken off its spindle to make sure that we have 100% in and out. So we've spent an inordinate amount of time. Luckily on this, we've got velocity sensor on the outside air coming in. So we know how much air is actually being pushed into the building in relation to the floor plate volume. Normally it's a minimum volume maintained by the dampers to suit the outside air quantity to suit the occupation of the building. So we've been looking at changing the BMS to make our life a bit easier with regards to not creating a very warm environment inside or very cold environment inside, whilst maintaining ventilation rates at a high level. Because we're dealing with a plant that can handle about 5 or 6 air changes of full outside air in the space, it's actually a very high ventilation rate capability. So we've instigated control regimes, both through occupied and unoccupied conditions. So it runs 24 hours, tries to maintain a certain temperature. The dead band in the space is widened in unoccupied space, in occupied times. And all in all, say that has been quite a challenge to get that BMS working properly with regards to making our life more comfortable for the people that are coming back into the office, but also to guarantee that we've got a ventilation rate that is COVID secure. So that is my mini presentation. It's not science, it's actually applying the science. So thank you very much.
No, thank you, Steve. That's exactly what we hoped to end on, just a demonstration of the reality that faces many people as they reoccupy offices. So that's extremely helpful. We are a few, 3 minutes ahead of time. I would like to now, we will have questions. I'm just sort of compiling a list of questions from the whole afternoon, or the whole second session, should I say. In the meantime, I would like to offer Adam Boys the chance to take us through the aerosol transmission website, which is obviously very relevant to much of this afternoon's presentations.
Great. Can you see that?
I can see that, and we can hear you well.
Okay, great. So these are just a few slides for those of you that aren't aware of the Aerosols and SARS-CoV-2 website. Really, they were prompted by meetings, these RAMP meetings, and then meetings that we were having in kind of in other venues via either the Doctoral Training Center for Aerosol Science or within the Aerosol Society. And there was kind of enough momentum that we thought we should seek to organize the literature and our knowledge of it in some way, so that particular scientists from different disciplines could kind of be abreast of what the latest developments were in adjacent fields. So that was our overarching goal, and we've made it somewhere towards that, and I'll talk a little bit about it. So the objectives of the portal are first to define the mechanisms of the viral spread relative to aerosol and droplets. We'd like to identify high confidence knowledge, so summaries of what the literature has said in terms of whether we know things well, and frame low confidence knowledge to prompt research or new studies that might elucidate key bits of information that we don't yet know. Our target audience for this is really people that would be participating in this webinar. So those skilled in the area of aerosols, disease transmission, immunology, air quality, physicians, fluid dynamicists, and the like. And we want to provide a literature database with common keywords and summaries that are somewhat coherent internally within this database and website. And we want to keep that database and website current as new studies are produced. That's no small feat, given the kind of exponential growth in publications in this area. And I think we were just about on top of it kind of before people went away for holiday, but I think we've kind of after the August holidays, I think we've got a large backlog that we need to kind of look at and dissect and kind of interpret for the site. And then we want to engage with international bodies to ensure that relevant data is being disseminated. We've had talks with colleagues in the US and Canada, and as a part of the European Aerosol Conference last week, introduced this there as well. So it is kind of getting known internationally. So how we've broken up the problem, so to speak, of aerosol spread is in a number of different mechanisms, and we're limiting ourselves to the transport via aerosols, where we feel like we have unique expertise. And so that's the expiration component, the transformation component, droplet loss, as well as aerosol generating procedures, droplet transport, and viral transformation. And then ultimately, airborne exposure via either physical transport or aerosol inhalation. And so those are the various mechanisms, and we've assigned editors, so to speak, and co-editors for each one of these. And many of the people that are on this call are serving in those capacities, but we certainly welcome any willing participants to this, because I think it is important that it's reflective of the diversity of the community that will be using and producing this information. So the website structure is it's a landing page. Currently, it's hosted by the Aerosol CDT. It will hopefully within the next few weeks move over to the UK Aerosol Society for long-term kind of use. So it's a landing page highlighting these mechanisms. Then sub-pages highlight the high certainty and low certainty knowledge for each one of the mechanisms. And then ultimately behind that is a database. So the idea is that the high certainty and low certainty pages give an indication of maybe seminal studies that have helped influence or shaped the knowledge, but there's a vast array of literature that will be available in the database itself. And that database structure will be hopefully useful for researchers and practitioners. If you're interested in participating in this, as I said, we're welcoming participants. They can be for somewhat niche kind of applications, or if you would like to kind of dive in and help more broadly, that's great. We are going to be using and have used the Aerosol CDT PhD students who are kind of growing in number, so they serve as good reviewers for the literature as well. And as I said, each one of those reviews gets a 100-word summary, so it's kind of our review of the literature, particularly kind of pulling out the mechanisms themselves. So if you're interested, there's a form here that you can click on or take a picture of the QR code that will take you to a site where you can indicate your interest in this project, and we'd be happy to have you on. The database as it stands is a Mendeley database that we're kind of manifesting on the website via BibBase. But what's nice about it is it allows you to sort by year or keywords. And again, we hope to have every publication kind of identified with a homogeneous set of keywords so that we can kind of interpret the literature well. And so this is kind of in its early stages and it's up and running. Feel free to go and have a look. And we hope to have kind of, once everyone's back and in full operation here in September, a number more studies to hit the website. A little bit about how we're just using this information as we bring the universe of publications in via the researcher app, which we monitor publications with fairly generic keywords. So hopefully we're kind of casting a wide net and then using editors to filter them out with the various mechanisms. Then we have co-editors or reviewers take and review the studies, write the 100-word summaries, and then update our high and low confidence knowledge on the front page facing web pages, as well as then deposit those publications and keywords. So that's all I wanted to say, just to introduce those of you who weren't aware of this resource to it, and certainly happy to take any questions or bring on any collaborators who'd like to help out with this. Thank you.
Thank you very much, Adam.